manufacturing of products. Product based on bacteriophages and other molecules. So what does that mean? In biotech or biopharma, it's quite typical that there are contract manufacturing organization or contract research organizations, so that help other companies, groups with manufacturing of products, and this is what we are doing. So just a second. So does that mean that we are at Jafral? It's to our knowledge is the only CMO CRO which primarily focuses development of production and production of phage-based products. So that means GMP and non-GMP. So for food industry, cosmetics, veterinary, and human therapeutics, I don't know, based on the description of Christophe, so based on the audience, GMP means maybe just additional introduction, explanation on that. So GMP means good manufacturing practice. So these are the products that can be used in humans. So in this stage for clinical trials, non-GMP means all other products, whether food industry, cosmetics, or veterinary. So yeah, we are unique from that perspective, that is our core business. So doing manufacturing, and because we are CMO, we don't have our own product. So it's like this, that our customers or with groups give us the phages, and then we produce them to whatever quality is required. With that, so what is then, let's say, the added value which we give to the phage community, the phage society? So that we have primary focus, that we have knowledge and quality system in place to work with many phage-based products. Also the facilities. So I didn't mention, but we are located in Slovenia, in capital city of Slovenia, which is Ljubljana. We have 2 facilities, and already while designing the facilities, we had in mind to that enables work with more phage products in parallel. So it needs to be in order to have a good quality product, it needs to be special designed to prevent cross-contamination stuff. So that was we had in mind during the building already, and as a CMO and CRO, so that from our work we give some synergies, and so that we develop the quality system set up at once, so that we have analytical methods set up once, also the materials and methods are set up, and we understand the critical process. So what does that mean in practice is if the groups or companies develop their own products, if everybody would needs to develop their own facility or the system or the process, it's quite expensive, and it takes a lot of time. So what we are doing is we learn from our work, from experience, and then that means that with every next project we also know more better, and which we can then help for all further products, so that we will hope also in the future with this added value, we try to then make the production manufacturing more efficient and also cost efficient, which at the end of the day is, I think, really important from the perspective that more groups, companies can afford either the clinical trials or different testings. More testing give more results, and to figure it out how this technology can successfully proceed to the good quality. So as said, location, we are located in 2 facilities. We have actually 2 different locations in Ljubljana. What does that mean? So because these facilities run 24/7, so all the temperatures, pressure, everything has to be monitored all the time. So if something happened with one facility, we still have on the other facility as a backup. Also what we call the critical material, which is starting material, stock material, or cell banks or phage banks, we have on 2 different locations, which again, from the risk management perspective, that means it's safer to have that if whatever catastrophe happens, we secure half of the material. Our products, like, so as I mentioned, they're phage-based products, but from the technical pharmaceutical point of view, we are producing cell banks, and phage banks that are starting material, then what is called drug substances. So that is, or in EU language also, APIs, pharmaceutical active pharmaceutical ingredients. So that is also monophages, and then if that is mixed together as a cocktail, which is called drug product, and then also placebo. From the facility point of view, we need and we have grade D, grade C, grade B, and grade A, which means that we can produce also technically sterile products, which can be then used also as a IV products. Since we are working with phages from 2011, we work with really number of different fields of application: human therapeutics, veterinary cosmetics, food agriculture application, diagnostic, as well as not only different phages, but different phages for different bacteria. Some of them are mentioned here: so Listeria, Salmonella, Pseudomonas, Staph, E. coli, Clostridium, Klebsiella, Propion, Enterococcus faecalis, and also there are some new ones. I have to say that it's always gives us additional interesting things or challenges, not just taking on new phage, but of course in taking on new bacteria, because it can be aerobic, anaerobic, they can grow differently. So also during production, that does influence the process. We produced more than 40 phage or bacterial banks. We produced more than 30 APIs for clinical trial materials. Produced 7 drug products that have been successfully used in clinical trials. Also, we produce several phage products for compassionate use. From production point of view, just maybe as an overview, what we are doing, what we can, how can we help to the phage field, is from production point of view that we can help from the start. So if somebody brings us the bacteria that caused infection, we can help with phage isolation, also the testing, the characterization, how that is, how good it is, or what is important then for development of such phage product. Then do the small scale process development, and in this scale, in this stages, also small scale production. That is usually also then whether tested in vitro, in vivo, small animal trials. What we always try to suggest is do all this testing also, put all these products also in stability as early as possible, because there are many products that maybe are good, but they are not stable. So it's better to test it sooner, and this cannot be speeded up. Then after the small scale, we also do the scale up, and with the scale up production, we also always test stability again. And then whether if that is the clinical trial material, produce the GMP clinical trial material, alongside, of course, again, the stability, or if it's non-GMP, non-human use, also do the routine production and then stability program. So this is the whole path. So as said, so we can help from start, and or we have also cases that somebody comes to us and say, we are just interested in the scale up and further, because we already did the initial part from scratch. So that is one part, and the supporting part, which the production cannot go further without the analytical matters. So, and also analytics, we have approximately 90% of analytical methods we are doing in-house, which is quite beneficial for us, since for the good process development manufacturing, it's good to have good analytics. So if we are developing that alongside each other, it goes really smooth. So, and we understand the process better. We start with analytical method development, then the qualification, following the method verification, validation, and routine usage. From that perspective, again, we try to do that there is some synergy between the projects, so it's not each product or project is, so it is then there is more cost efficient, that because we have most of the QC methods already developed. So what we only need to do is to do optimization per each product. So we don't need to do development from scratch, which is more cost efficient. Also from time-wise perspective, it's faster. Practically manufacturing capabilities. So we are doing early process development in laboratory scale. What we say is laboratory RLMI flask. So it's 100 milliliters, half liter upstream, downstream. Then small scale verification, 1 liter scale to test robustness and repeatability, process capability, stability testing. Then is scale up, what we also call it's engineering runs, so large scale production, upstream, downstream, scalability testing, and then routine scale production, large scale, and the production of required material. At this stage, we have, that is per one batch, we have the scale is 30 liter, the upstream, but with the products that we produce so far, that was sufficient for phase 1 or phase 2, or even would be more. So it really depends on the specification or the application of the phage product. This is just example, just technically. So what we are doing, what we call a critical process parameter, defining, so for upstream process, so how you grow phages that we test. So which parameters are important. This is from perspective of efficacy, so that we can produce like a product like efficient, but also other thing is from GMP perspective, you have to understand which parameters are important, so that how you control the process. So that is done. And then for the downstream, which is purification, which is from the safety of the patient point of view, maybe even more important. So how do you achieve sufficient purity? And I have to mention here that we are using all the pharma grade chemicals, so that the chemicals that are scalable, try to avoid centrifugation, also cesium chloride. So technically what our raw materials that we are using, it's what is also otherwise used for other biopharmaceuticals. So they're compliant, GMP compliant, and recognized as safe. And in this case, there are different ways always how to get the good quality, the final purity of the material. Now it just really depends whether that is oral product or topical or IV. So then we are also playing which steps, so which methods has to be used at which step to achieve the best quality in the best yields. Then from the analytical capability point of view, what we have is analytical capabilities can be in different stages. So what we call it in process. So during the production, we are doing analytical methods so that we understand whether the process went as we expected. Then the analytical capabilities as a release method. So once the product is produced, that we tested whether it meets all the specification, stability products. So testing stability, and also then whether if it's already some testing was done on the field trial or animal trial, so that we can, if they are sending us samples back, we can evaluate the specific parameters, what we can do. So whichever methods we have in house. So here are just few example of the methods. So plaque assay, so that's, let's say, main method is potency. Then also bacterial counts, endotoxins, identity PCR, qPCR. So again, some of the stuff are really, let's say, generic between the different phage products. In some cases, we have to do optimization or do part of the development, as in this case for the PCR or for the qPCR. Testing for the impurities are proteins, as the page ELISA, and then also for the DNA. So agar gel DNA measurement as total or as a qPCR method. Some of this method we can also, again, as said, we do outsource as a ddPCR or electron microscope. So as part of the characterization, also sequencing. And with this all the analytical methods, we first evaluate the quality and then also understanding the process from the yield or the quality perspective. And then this is maybe just out of interest how final product can look. I would say that many or majority of the products, phage products, are with the titer 10 to the 8, and then different purity depends on the application. There are some products, phage products, that they want to have really high concentration, more than 10 times 10 to the 12. In this case, it's, as you see an appearance on the left side, the final product is quite opalescent in that case, but still can be filtered to 0.22, and then can be evaluated with the electron microscope, with the HPLC system. So different techniques to assure the good quality of the product. And then last but not least, as said, it's really important not just to have the product, but to have the stable product. So here are a few just examples. If you see on the slide, it's phage A, like 3 graphs of the phage A, where we tested different buffers at different conditions. So the top left is 2 to 8 degrees. So for example, 4 different buffers, phage was stable for all of them. Then we have on the top right, it's 25 degrees, where we already see that buffer 4, it's not stable for, it's not good for one phage. And then on the bottom right, it's 40 degrees, where that effect is even higher for the 40 degrees. And then the same formulation as you can see for the phage B below on the left, it's only 1 buffer. It's okay. So what I'm trying to say with that is that there is not some general rule which buffer will definitely work. So there are some buffers that are preference, but still it's always need to be tested, the monophage or drug substances, and that is always what we are doing before we are mixing them together, so that we understand how they behave. And in recent, so in last year, we also worked or start to work with on the spray drying, so that we can also prepare the final formulation is actually a powder, so that we do the spray drying of the phages. And here are just 2 graphs of different parameters that needs to be that does influence on the efficacy of the spray drying. And yeah, that's all from my side. So thank you for your attention, and if there are any questions, I'll be happy to answer.
So thank you for this great presentation, a great applause from all the audience. We already have a few questions. The first one is from France Fan. The question was about the shelf life of your product, and if I may add, about the stability. If they are spray dried, do you need to store them at a specific temperature?
Yeah, maybe first for the spray drying. In general, with the spray drying, the intent is that you can easier store it at 25 room temperature. So otherwise, most of the phage products in liquid, we are storing it 2 to 8. So with the spray drying, it's more or less what we are looking at the moment, is that would be maybe more also for some animal product or something like that, so that because you cannot afford to have 2 to 8. And then the other option would be, of course, if you are spray drying, that you can also then do the capsules along the way. So that is first. And the second one, what is the shelf life? So in general, we are doing, as said, whether us or our partners, whoever gives us the product, some selection of the phage with good stability, and that for us means that it's in general like stable for 1 year for sure. So then it's only longer. there's always some compromise where some phage can be really good, and it's not really so stable for 1 year. But I would say that is some at least starting point. So around there.
Okay, thank you for your question. We have another question from Gregory Resch. Gregory, I give you the word.
Yes,
Hey,
hi,
Gregory.
Frank. Thanks for your talk. So I have a question, Frank. I'm not sure you said it or I got it. Maybe I didn't get it. When you say that you produce phages in GMP or non-GMP, it means that you have an official certification for GMP production of phages, which is recognized at the EMEA, and we can find it under the EUDRA GMP website, for instance?
Correct, yes. Yeah. So it goes like this, that it's for each country in Europe is the same. So our national authority audits us and audited us, and then it gives you, so for whatever product, so in our case for bacteriophage production, and then gives you a certificate which is then valid for the whole Europe. So that is within the EMEA. And then and that also gives you the opportunity that you can also ship to FDA, to US or Australia or something like that. So yeah.
Okay, great. Thanks.
We have another question from France Fan about the TEM and the analytical tests that are performed. Can you talk a bit more about this?
Yeah. So which methods can we do it?
About the analytical method and the TEM test.
Yeah. So for TEM test, actually we don't have that one in house, but we have actually one institute here in Slovenia, which we work regularly with them, which they are also expert in the viruses and also as well also in the phages. So that is something like really regular characterization that we are doing. So for each product we are sending them for analysis. So I hope that's enough information. If you want more, or just let me know. And yeah, for all other analytical methods as mentioned, so we are able to characterize from the impurity point of view or from the product point of view the methods that were listed there. And we are always open. Also, if there's some other methods, and that is what we are seeing, that there's also more now, let's say GMO phages, where maybe they want to do some other analytical testing, whether testing for specific gene that is still there, or maybe some antigen or whatever protein on the envelope, that method needs to be developed. So that is also quite regular work for us. So yeah, also developing some new methods.
So thank you for this information. Is that enough for you, France Fan?
Hello. Yes and no. My question was, for which purpose do you use TEM? Is it to check purity, odor? Because you, as far as I understand, you are using such method as a kind of QC test, or it's only to know the morphology, or is it for another purpose?
Good. So in general, as a QC, so now I understand. Thanks. So it's for morphology. That is at the beginning, but we also, as a in process testing, there are also cases where we are testing. I don't know if we are seeing some kind of unexpected degradation or loss during the process. We go and check for the TEM, and because we can see actually there's some kind of degradation, or I don't know, some aggregation, something like that. So that is the other more process point of view that we are using.
Okay, thank you, Frank.
So we have a last question from the audience before the next speech from Cristina Maria Manon. I give you the word.
Yeah, thank you for this interesting talk. I was wondering what the average times are from, let's say, somebody comes with the phage they want purified on a small scale to have the final stable product produced by you.
You mean that would be for non-clinical material or clinical material? Both.
Yeah, either
Okay.
both. Yes, I guess.
Okay. Yes. So for non-clinical material, so yeah, actually, it's a good question. So that I can also explain. So our business model is different to majority of the, let's say, GMP or whatever CMOs. We do always, and because we start like this, we take the project already from the early stages. So we have lots of projects that somebody found one phage, or they're doing like really small scale, and they give us, and we do early production testing, and then we prepare that for animal trials, and then can be turnaround time can be 1 month or 2 months. So it's really fast, and then depends how they're improving, we can then wait or so whenever they came back, and we move forward. In order to so just to do that optimization can be 1 to 3 months. That can be really fast. Of course, then whether if it's stable or not, then it's a factor which we cannot speed up a lot. So we have to wait for stability. But let's say like this, that what we typically do is, if we produce that in 2-3 months, then waiting for stability again 3 months, whatever it's that you at least get some sense about that. If we want, let's say, from start of the clinical project from zero to the clinical trial material, it again depends whether that is one phage, or maybe typically for the DP it's 3 to 4. I would say it's approximately 1 year that it would be the clinical trial material. So from the start of the project till the end. So that's really rough.
Okay, thanks.
Welcome.
So we still have 2 questions, and then we will skip to Austria from Arshana. What is the rate of stability for Staphylococcus aureus with the 4 buffer that you talked about?
So yeah, that's again, it's really tough to say, because it's not really depends so much of the buffer. It is as well, but if there's not stable staph phage, it's really hard as well. But nevertheless, so the staph phages in general are quite stable. So I would say they can be achieved like 1.5 to 2 years with 0.5 log decrease, which is completely fine. So they in general quite stable. 1.5 to 2 years.
Thank you. And from Lucille Plourde, we have the question about what is the objective of sequencing testing, identity of stability?
So what is the sequence of testing?
The objective, yes.
The identity would be, we are always doing so whether we are doing the sequencing and then maybe the PCR for identity, but then on stability, it's actually the potency, the major factor. It's not actually the identity wouldn't be if you have good label, stuff like that.
Okay, thank you a lot for all this explanation and the great presentation you gave us. Great round of applause from you, Frenk, and thank you.
Thanks.
So we are coming to the second presentation for tonight. The second presentation is going to Vienna with Lorenzo Corsini, which is the CEO and co-founder of Phagomed. And we give you the presentation to the word to talk about more about Staphylococcal aureus.
Thanks very much. Thanks very much for inviting me. I'm going to share my screen. Just a second. Okay, can you see it? Does it work?
Yes, everything is fine.
Okay. All right, I'm Lorenzo Corsini. I'm the CEO and co-founder of Phagomed. We're a biotech company in Vienna, and we develop targeted antimicrobials for treating chronic and persistent diseases based on both bacteriophages and endolysins. So just to give you an overview of what we do and where we are from the science, we have a lysin prediction platform. We work on gram-positive and gram-negative lysins, have candidates for either. And we've got these epsilon square phages, which are superior bred non-GMO phages. And today I'm going to mostly talk about these phages because this is a phage audience. We have two drug candidates. One is PM477, which is an endolysin specific to Gardnerella bacteria, which we develop for the treatment of recurrent bacterial vaginosis, the most frequent vaginal infection in women of reproductive age. And then we have PM399, which is a cocktail of 3 epsilon square phages specific to Staphylococcus aureus, and which we develop for the treatment of implant-associated infections. Obviously, we want to bring these products to the clinic. We're 15 scientists in total. We have a big lab in Vienna. And today I'm going to show you a little bit about the details of what we're doing. So the problem that we started to work on, that we wanted to solve, is that, in our opinion, most wild-type phages are consistently unreliable. This is obviously not surprising in that phages didn't evolve to eradicate bacteria to be used as pharmaceuticals. They just want to propagate themselves. And to illustrate the problem, you see on this slide, for the example of Staph aureus phage 812, which is a well-known and published Heraldviridae phage, how it works on 3 different Staphylococcus aureus strains of 3 different clonal complexes. And see, we made the experience for this phage, sorry, but also for most other phages, that while the plucking typically works well, and there is a broad plucking host range on a broad range of bacterial strains within the species Staph aureus, these wild-type phages perform less well in some other characteristics. For example, growth in blocking bacterial growth in suspension, or also biofilm assays. And so with this example, you see there are 3 different strains. On the top one, the phage is able to fully block the bacterial growth also in suspension. The second strain looks equally well by plucking, but after 9 to 12 hours, you've got resistant outgrowth. And in this third strain, even there's just barely any reduction in the bacterial growth. So the question is really, for these 2 bottom strains, would this work in the clinic? And a bacterial infection in a human, is it more like this, like the plucking, where you see nice plucks with a nice efficiency of plucking, or is it more like here, and you would get fast resistant outgrowth? And in this case, would this be any advantage for the patient? We analyzed this in much more detail. For example, we calculated these host ranges on 110 bacterial strains, which had a very controlled diversity, meaning the composition of this panel reflects the epidemiology in humans. We measure plucking and kinetic host ranges, which is, so the kinetic host range is defined by us as the ability to block bacterial growth in suspension. And you see that bottom line, there is this big gap. While most phages have a very broad plucking host range, the kinetic host range is very low. This chart on the right shows that there is very little correlation between the ability to block bacterial growth and suspension and the efficacy of plucking. And you can read all the details in the paper that we recently published. But bottom line, we felt that it would be pharmaceutically much more useful to close this gap and have phages that are lytically more aggressive, that have increased virulence over the wild types. So we wanted to improve the host range, we wanted to improve the virulence, and we wanted to reduce the propensity of resistance formation. And to do that, we chose to induce random homologous recombination, sometimes also called the Appelmann's protocol, which is essentially inducing phage sex upon superinfection and inducing basically recombinations between phages in a way that then you can select for beneficial properties and improve the phages of the wild types. And just 2 words on why we chose this method over other methods. Obviously, there is, as an alternative, there is for example, chemical mutagenesis or sequential training. This is less efficient in a way because you just accumulate in a linear way the mutations and don't combine the potential existing advantages from different ancestors. There's a method of generating tail spike or libraries of the tail spike protein, which also works, has its advantages, but you're generating GMOs, and also, as I'll show in a moment, host range and resistance is not only determined by the tail spike proteins, and so here you would be working on just one. Similar for CRISPR payloads, you're generating GMOs, and you don't solve the problem of adsorption resistance or the narrow host range of wild type phages. So all these methods obviously have their benefits, and I'm sure I didn't include all the methods here. We focused on random homologous recombinations. And this was very successful. So as you see on this chart, and you can also read it in our paper, we virtually doubled the kinetic host range of the wild type phages. So here we generated intercrossings between phage 2002, 812, and BT3. These are wild type phages. 2002 is a phage that is somewhat similar to phage ISP. It was isolated by Greg Resch's group in Lausanne. 812, we just talked about, it's a well-published phage. And BT3 is a phage that was isolated at DSMZ in Germany. And the best ancestor, which is a genetic recombination of these three, as I said, has a double as high kinetic host range. So again, this is the ability to block bacterial growth in suspension. We generated also some other epsilon square phages, as we call them. So this is for evolution squared, accelerated evolution. When we combine these three phages, PM4, PM93, and PM56 into a cocktail that we call PM399, the cocktail has a kinetic host range of 92%. We also reached quite significant improvements in the plucking host range for the epsilon square phages. And you see that for the cocktail, it has a plucking host range of 93 and a kinetic host range of 92, which means essentially we close the gap between the kinetic and the plucking host range, the problem that I showed you at the beginning. So obviously this will have some, or these are some very useful pharmaceutical properties for a drug that is supposed to be used on many different patients in a reliable way. We also analyzed this in much more detail. This chart, or this shows, this is a series of kinetic curves recorded in a 96 well plate, and you see here that phage PM4, which is the best epsilon square phage, is able to eradicate or fully block the growth of all of these 11 strains here, and for several ones, none of the ancestor phages was able to even just suppress the growth. We analyzed it in even more detail to characterize the actual virulence of the phages following the recipe in this paper here, Storms et al. 2020. Essentially here, we measured the kinetic of these phages with different starting MOIs on a number of bacterial strains. And as you see here, phage PM4 is able to fully suppress the growth of this strain at a 2 log lower starting MOI compared to the best ancestor 2002. And this can be evaluated more systematically, calculating this virulence index and these virulence curves, which are basically the virulence index as a function of starting MOI. And as you see here, the dashed line is PM4. It has a higher virulence than the wild type phages, especially at the low MOIs, which is critical to be able to employ this phage also at lower concentrations. So the epsilon square phages are mosaics of the ancestors, and this is shown on one example for one region of the genome for phage PM4. This is PM4 has roughly 150,000 base pairs, and this graph shows the last 20,000 base pairs. And this is an alignment, obviously PM4, and then you have the ancestors where gray means identity to PM4 and black means difference to PM4. And as you see here, this part of the long terminal repeat is identical to the ancestor phage 2002. Then you apparently had a recombination site, and then the sequence continues as BT3, changes back to 2002, changes back to BT3, back and forth a couple of times. Sometimes there are even insertions of 802. So this is a mosaic. The process behind this is obviously a natural process. We know that most phages are mosaics of some ancestors, but by selecting for properties that we regard as humans, as a company, as pharmaceutically relevant, we're able to generate non-natural phages that are non-GMO, but also that are clearly superior to the ancestors, and that are patentable also, unlike any other phages, because these combinations allow basically thinking about patents in a completely different way. We also looked at resistance formation, and we used two different methods here. One is spontaneous resistance formation, where we created a cell suspension of 10 to the 11 cells and just measured if we would find even one resistant cell. This was negative for the components of PM399 and for the cocktail itself, which is very useful because in an actual patient, you can calculate that it would be very improbable to have 10 to the 11 bacterial cells, meaning that it would be extremely unlikely for a patient treated with a cocktail to develop even one resistant cell in his body. We also tried at least to generate resistance formation by passaging with submic concentrations, and we're not, sorry, a bit of a cough. We're not able to generate resistance, while in comparison, the standard of care antibiotics for MRSA, rifampicin, but also vancomycin, for rifampicin, we can generate full resistance beyond the limit of detection after 1 of 2 rounds. And also for the combination of vancomycin and rifampicin, we can significantly increase the MIC after actually a small number of passages. We also tested the biofilm host range on a reduced panel of bacteria, this times 10, of again, different clonal complexes. And again here, we see more than 95% reduction of the biofilm CFU in more than 80% of strains, which is very useful for the application of implant-associated infections. As a comparison, we also measured the biofilm eradication potential of vancomycin and rifampicin at super high concentrations. It's not possible to eradicate these biofilms, even at concentrations that would be much higher than what can be used in humans. And this is again reflecting the observation that you cannot treat an implant-associated infection with antibiotics alone. We also tested this in vivo with a Zimmerli model. This is I would say, the most rigorous animal model for implant-associated infections, where you essentially implant these Teflon cages under the skin of guinea pigs. Then the cage is infected transcutaneously. We wait for 3 days without any treatment to allow a strong biofilm to form. Then there is a 4-day treatment regimen, then a 5-day washout to allow potential residual biofilms to grow back. And then the primary endpoint is sterility of the implants after explantation of the cage. And the result is what you see here. In blue is non-sterile, in green is sterile. Buffer control, 0 sterility, antibiotics alone, 0 sterility. Whereas with the phages, you have a dose response. And with this regimen, we had 30% sterile cages with epsilon square phages in a higher dose and more frequent administration. We had even 67% of sterile cages, which is quite a breakthrough, because it's well known that implant infections cannot be treated with antibiotics alone. You need surgery all the time, mostly an actual exchange of the prosthesis. And so being able to, at least in some cases, fully eradicate the infected implant without any surgery indicates that it might be possible to use phage therapy to improve the probability of success of such interventions, and also with much less invasive surgical procedures. So to summarize, we have a very potent and very robust cocktail. We have a very broad kinetic and plucking host range, very strong biofilm performance, no resistance formation. They're active in vivo. Also from a manufacturing perspective, they're very good to handle. So we can achieve in shaking flasks 10 to the 11 PFU per ml. Which means that in fermenters you could achieve even more. We purified them from prophages. They're all propagated on prophage-free strains. We have them purified and sequenced, and there are 2 patents pending on the cocktail. So Phagomed focuses, as I said in the beginning, on bacteriovaginosis. We have a number of other licensed candidates, and regarding phages on periprosthetic joint infections. And as a still small biotech company, we can't develop this for all the breadths of potential indications where these phages could be useful. So we're also looking for partners to maybe co-develop PM399 and the epsilon square phages in other indications. We also did a similar trick with Staphylococcus epidermidis phages. Also published a paper on that one. So, and with that, I would like to thank our fantastic team in Vienna who did all the experiments and produced all these results. And if you're interested and want to hear more, please reach out. And thanks very much for your attention.
Thank you for this nice presentation. We already have some questions from the public. I give the word to Varun Vakabeshbavda. You can maybe read your question out. The question is, while you see that both planktonic and biofilm condition are different, and we might see different phage infectivity in both conditions. Did you find any such case in your study? Or do you choose only those phages showing the highest activity in planktonic culture?
So we chose them by planktonic culture, but we see a clear correlation. So especially, I mean, as you can imagine, in a biofilm assay, a phage that allows resistance formation after 9 to 12 hours, you won't see any effect in a biofilm assay because it will just grow back. We are right now testing in animals the difference also between wild types and the epsilon square phages. But in general, we believe that seeing this much higher virulence and a broader host range and lower resistance formation, this is a very clear sign that the phages might also be more active in a clinical setting.
Thank you. Maybe some other questions? We have a question from Jonas Koch. You emphasize that these phages are non-GMO, which is debatable in my mind. In this non-GMO label important, is this non-GMO label important for regulatory approval on the long run or rather for marketing?
No, this is I think makes a big difference in the regulatory approval, especially in Europe. I think probably less so in the United States, but in Europe you will have problems to release a genetically modified organism into the environment in an uncontrolled way, which you would have to do after treating a patient. If you don't want to autoclave the patient, the phage will go into the environment. so you think it's debatable? Well, we discussed it a lot with our regulatory consultants. I think there is quite some consensus that this will not be GMO, also because the process by which this is happening is a natural process. If you look at the genomes of wild-type phages, there are all mosaics in one way or the other. So have occurred by recombination with some other phages. So this is quite a natural process.
And if I may ask a question about this, is the marketing for the GMO or non-GMO something important, or do you have no feedback about the customer, the physicians that want to treat only or only without GMOs?
I think this would depend a lot on the indication. A lethal, potentially lethal, or really life-threatening indication like a sepsis, like a implant-associated infection, I don't think many people would have a lot of questions around is this GMO or not. But in other settings, let's say maybe a urinary tract infection or some skin disease or something like that, there might be questions around that might be a disadvantage, at least in Europe. We don't know. I mean, nobody really tested that, but I could imagine this.
Thank you. And we also have a follow-up question. If you claim that they are created by a natural process in a gentle nudge, are the phages still patentable? Chances are that recombination may have happened already.
Yes, of course, but you can, well, as long as this has not been published, this counts as has not happened. So we could show that this is novel just by looking into the databases, and then there is also a way of patenting this.
So another question from Arcana. She's asking about the concentration you use for biofilm treatment.
We test. I mean, we test all different concentrations. Also, there we work with different starting MOIs, really ranging between 0.1 up to 10 or 100.
Thank
you.
you.
It's really important to test the dose dependence and not just test one dose.
So I give the word to Fabien Labroussa.
Yeah, hi, can you hear me? Yeah.
Yes, we can.
Okay, so, I was just wondering how much diversity you can generate using that homologous recombination, and what was the selection criteria you had, you know, at the end of the process, because I guess you can really generate a lot of diversity, you know, at the end of the homologous recombination. So what is like the you know, really the criteria or the, you know, the parameters you checked for selecting those phage at the end of the process?
Yeah, so there were a number of iterations, and there were a number of different selection filters. So we tested for or selected for basically activity and no resistance on a number of very diverse bacterial hosts, but also for activity under physiological conditions. And there are many different selection filters that can be applied.
Okay. And is there a way to accumulate, you know, virulence genes or something like that can later on be problematic on such phages?
Of course, because these are strictly non-temperate phages, but of course they were exposed to prophages and that are just on the genomes of the host bacteria that we use. So yes, there was a risk, but we sequenced the phages and this was not the case.
Okay, thank you.
Thank you for the question. And we have another question from Christina Maria Manauer.
Yes, thanks for this great talk. I was wondering, do you think your protocol or method is applicable also to phages for other bacteria like Pseudomonas aeruginosa that's also notably bad in biofilm formation?
Yes, I mean for Pseudomonas, like the academic version of this was already published, and yeah, I mean there are many ways of improving these breeding techniques. Yeah. So I think in principle this can be applied with any phage.
Okay. Thank you.
We have a follow-up question about the patents from Varun Bavda. The question is, but how can you call that wild type and patent? To my knowledge, we cannot publish wild type strain.
This is not wild type. This is not wild type. This is not a phage that was found in nature. We generated it. It's a bit similar to you know, cherry tomatoes. They're very different to the tomatoes that somebody found in some forest a couple thousand years ago, and you can also protect the intellectual property on crossbred plants, and this I think a bit homology here to the phages.
Thank you for the answer. J'ouvre aussi la discussion en français. Si une personne a envie de poser une question en français ou avoir des Détails en français, ich eröffne auch die Diskussion für die Leute, die auf Deutsch eine Frage stellen wollen, sei es auf den ersten oder auf den zweiten Talk. Les questions peuvent être en français sur le premier ou le deuxième sujet. It seems not to be the case. Also, thank you a lot for this great talk and for all this information. It was a pleasure to have you both of you for this webinar. And thank you all of you for participating and for the nice questions. You receive a round of applause from all the audience. Thank you a lot.
Thanks for inviting us, and thanks everybody for listening.
Yeah, thank you.
And we are coming to the end of this June webinar. The next webinar will be at the beginning of September. All the persons that were interested in phageSuisse will receive an email with more informations. We have a few topics that are now in discussions. I cannot give you some more information right now because we are still in discussions. The next event for phageSuisse will be in 2 weeks. In the 15th of June, we will have the General Assembly of phageSuisse. All the members will receive an invitation for the next General Assembly, and we can't wait to see you all present for this event. Thanks a lot. And thanks a lot again for the two speakers for the nice presentation and interesting thought that you brought us. Merci à toutes les participantes et tous les participants pour votre participation. On se réjouit de vous revoir pour la prochaine organisation, pour le prochain webinaire qui sera en septembre et pour l'Assemblée générale qui aura lieu dans 2 semaines. Les membres recevront l'invitation tout bientôt. Danke auch an alle Teilnehmerinnen und Teilnehmer. Der nächste Event ist die Generalversammlung, die in 2 Wochen stattfinden wird, und der nächste Webinar wird im September stattfinden. Vielen herzlichen Dank. Thank you and see you soon.
Bye, have a nice evening.